Enzymes and vitamins. Enzymes general features. An enzyme is a biocatalyst that increases the rate of chemical reaction without itself being changed in the overall process. Enzymes have several characteristics that make them unique. Enzymes exhibit enormous catalytic power. Enzymes are highly specific. Most but not all enzymes are proteins. Enzymes can be divided into two general categories, like simple enzymes, which consist entirely of amino acids, and conjugated enzymes contain a non-protein group called a cofactor, which is required for biological activity. Removal of the cofactor from a conjugated enzyme produces a simple enzyme called an apoenzyme, which generally is biologically inactive. The complete biologically active conjugated enzyme, simple enzyme, plus cofactor is called a holoenzyme. A cofactor can be linked to the protein portion of the enzyme either covalently or non-covalently. Some cofactors are simple metal ions and other cofactors are complex organic groups which are also called coenzymes. Cofactors which are tightly associated with the protein covalently or non-covalently are called prosthetic group. Vitamins and their coenzyme forms. Vitamin B complex, thiamine B1, thiamine pyrophosphate, decarboxylation aldehyde group transfer, riboflavin B2, FAD and FMN, redox reaction, riboflavin and the pyridoxine B6, pyridoxal phosphate amino group transfer, nicotinic acid niacin NAD plus and NADP plus redox reaction, pantothenic acid, coenzyme A, biotin, biocytin, folic acid, tetrahydrofolic acid, 1 carbon group transfer, and folic acid, then vitamin B12, deoxyadenosyl intramolecular rearrangement, and ascorbic acid, vitamin C, that hydrolyzes everything. The vitamins in the human diet that are coenzyme precursors are all water-soluble vitamins. Some enzymes and their cofactors Fe2 plus or Fe3 plus, cytochrome oxidase, catalase, peroxidase, xanthine oxidase, cytochrome oxidase, lysyl oxidase, superoxide dismutase, carbonic anhydrase, alcohol dehydrogenase, carboxypeptidase, hexokinase, enolase, glucose 6-phosphatase, arginase, enolase, pyruvate carboxylase, pyruvate kinase, urease, denitrogenase, and xanthine oxidase, along with the glutathione peroxidase. Classification of enzymes. The International Union of Biochemistry instituted a systematic naming scheme for enzymes. Each enzyme is now classified and named according to the type of chemical reaction it catalyzes. The following are the 6 major enzyme categories. Oxidoreductase. Oxidoreductase catalyze oxidation reduction reactions. Subclasses of this group include the dehydrogenases, oxidases, reductases, peroxidases, and hydroxylases. Then we can see an ethanol, CH3CH2OH, transforming into acetaldehyde with the help of alcohol dehydrogenase. NAD plus transforms to NADH plus H plus. Next is the transferase. Transferase catalyze reactions that involve the transfer of groups from one molecule to another. Examples of such groups include amino, carboxyl, carbonyl, methyl, phosphoryl, and acyl group. Common trivial names for the transferase often include the prefix trans. The groups include amino, carboxyl, carbonyl, methyl, phosphoryl, and acyl groups. Examples include the transcarboxylase, transmethylase, and transaminases. So D-glucose transforms to D-glucose 6-phosphate with the help of hexokinase. Next is the hydrolases. Hydrolases catalyze reactions in which the cleavage of bonds is accompanied by adding water. The hydrolases include the esterases, phosphatases, and peptidases. So when we add water into the C-terminus of polypeptide, shortened polypeptide is produced and C-terminal residue will be produced. Then the lyases. Lyases catalyze reactions in which groups are removed from a double bond or are added to a double bond. That is the function of lyases. Decarboxylases and synthases are examples of lyases. Pyruvate decarboxylase, acetaldehyde is formed. Then the isomerase. This is the heterogeneous group of enzymes. Isomerase catalyze several types of intramolecular rearrangements and yield isomeric forms. The epimerase catalyze the inversion of asymmetric carbon atoms. Mutases catalyze the intramolecular transfer of functional groups. So malate will be there. Malate isomerase transforms it into fumarate. Then ligases will be there. So ligases catalyze the formation of C, CS, CO, and CN single bonds. The energy for these reactions is always applied by ATP hydrolysis. The names of many ligases include the term synthetase. Several other ligases are called carboxylases. Pyruvate undergoes ligation. Ligases are there. So pyruvate carboxylase transforms it into oxaloacetate. The enzyme commission has given each enzyme a number with 4 parts like EC2712 hexokinase. 2712. Hexokinase is named as 2712. The first 3 numbers define major class, subclass, and subclass respectively. 2712 is the class number of hexokinase. So 2 will be the major class, and then the subclass and the subclass respectively. The last is a serial number in the subclass indicating the order in which each enzyme is added to the list. Major class, subclass. Then the order in which last number, that is a serial number in the subclass indicating the order in which each enzyme is added to the list. Alcohol dehydrogenase. Common name and enzyme commission number of some enzymes. Alcohol dehydrogenase is represented by EC1111. Alcohol dehydrogenase. Phosphofructokinase and acetylcholinesterase 3117. 3117 is nothing else but the acetylcholinesterase. Alcohol dehydrogenase is numbered as EC1111. Alcohol dehydrogenase and acetylcholinesterase 3117. Major class, subclass, subclass, and the serial number in the subclass indicating the order in which each enzyme is added to the list. Next, alcohol dehydrogenase and acetylcholinesterase. Okay, what enzyme does? A chemical reaction between two substances occurs only when an atom, ion, or molecule of one collides with an atom, ion, or molecule of the other. Only a fraction of the total collision results in a reaction, because usually only a small percentage of the substance interacting have the minimum amount of kinetic energy a molecule must possess for it to react. When the reactants collide, they may form an intermediate product whose chemical energy is higher than the combined chemical energy of the reactants. In order for this transition state in the reaction to be achieved, some energy must enter into a reaction other than the chemical energy of the reactants. This energy is the activation energy. The enzyme lowers the activation energy of a reaction, thereby increasing the fraction of molecules that have enough energy to attain the transition state and making the reaction go faster in both the directions. However, the catalyst does not change the relative energies of the initial and final states. The free energy of reaction, delta G, remains unchanged in the presence of a catalyst, so the relative amounts of reactants and products at equilibrium are unchanged. So in the presence of a catalyst, relative amounts of reactants and products at equilibrium are unchanged. In other words, the catalyst does not influence the position of equilibrium. It increases the rate of reaction by lowering the activation energy. So, reactants will be there. We can see reactants are there, then products are there. So, reaction coordinate will be represented, and then the activation energy without a catalyst is higher, and activation energy with the catalyst is lower. How enzyme operate? There will be an active site. Active site of an enzyme is the region that binds the substrate and converts it into the product. The substrate is bound in the active site by multiple weak non-covalent interactions like hydrophobic H bond, ionic interactions, etc. Active site of an enzyme is the region that binds the substrate and converts it into the product. The substrate is bound in the active site by multiple weak non-covalent interaction like hydrophobic H bond, ionic interaction. In the first step in catalysis, the enzyme binds to the substrate, the compound to be catalyzed. We represent the binding of the substrate to the enzyme by the equation E plus X is ES, where E is the enzyme, S is the substrate, and the product ES is the combined species that is called the enzyme substrate complex. This binding is highly specific. A given enzyme usually binds to only one kind of substrate. The region of the enzyme that binds the substrate molecule is called the binding site. In the second step, the substrate is converted into product, where EP is the complex of the enzyme and the product. Formation of the enzyme substrate complex. Two important models have been proposed to describe the binding process. The first, the lock and key model, assumes a high degree of similarity between the shape of the substrate and the geometry of the binding site on the enzyme. Second model takes into account the fact that proteins have some three-dimensional flexibility. According to this induced fit model, the binding of the substrate induces a conformational change in the enzyme that results in a complementary fit once the substrate is bound. The bounding site has a different three-dimensional shape before the substrate is bound. When the substrate is bound and the transition state is subsequently formed, the bonds are arranged. In the transition state, the substrate is bound close to atoms with which it is going to react. As bonds are broken and new bonds are formed, the substrate is transformed into product. The product is released from the enzyme. Catalytic mechanism. Enzymes commonly employ one or more of the following strategies to catalyze specific reactions. First is the covalent catalysis. The process of covalent catalysis involves the formation of a covalent bond between the enzyme and substrate. The modified enzyme then acts as a reactant. After the completion of reaction, the enzyme returns to its original unmodified state. The process of covalent catalysis involves the formation of a covalent bond between the enzyme and substrate. The modified enzyme then acts as a reactant. After the completion of the reaction, the enzyme returns to its original unmodified state. The proteolytic enzyme chymotrypsin provides an excellent example of this mechanism. Then, general acid-base catalysis. In general acid-base catalysis, a molecule other than water plays a role of protein donor or acceptor. The ionizable functional groups of amino acyl side chains and of prosthetic groups, if present, contribute to catalysis by acting as acids or bases. For example, chymotrypsin uses a histidine residue as a base catalyst to enhance the nucleophilic power of serine. Chymotrypsin uses a histidine residue as a base catalyst to enhance the nucleophilic power of serine. Then metal ion catalysis. Which are the major types of catalysis? Covalent catalysis, general acid-base catalysis, and metal ion catalysis. So metal ion can function covalently in several ways. For example, a metal ion may serve as an electrophilic catalyst, stabilizing a negative charge on a reaction intermediate. A metal ion may serve as an electrophilic catalyst. It serves as an electrophilic catalyst, stabilizing a negative charge on a reaction intermediate. The metal ion may operate a nucleophile by increasing the acidity of a nearby molecule, such as water, in the hydration of CO2 by carbonic anhydrase. The metal ion may bind to substrate, increasing the number of interactions with the enzyme and thus the binding energy. Enzyme kinetics. Let's learn about the enzyme kinetics. This is a major topic. Enzyme kinetics is the quantitative study of enzyme catalysis. Kinetic studies measure reaction rates and the affinity of enzymes for substrate and inhibitors. Kinetics also provides insight into reaction mechanisms. In a reaction of the form A plus B gives P, where A and B are reactants and P is the product, the rate of reaction can be expressed in terms of either the rate of disappearance of one of the reactants or the rate of appearance of product. The rate of disappearance of A is minus delta A by delta T, where delta symbolizes change. Concentration of A is the concentration of A in moles per liter and T is time. Likewise, the rate of disappearance of B is equal to minus delta B concentration by delta T, and the rate of appearance of P is delta P by delta T. The rate of the reaction can be expressed in terms of any of these changes because the rates of appearance of product and disappearance of reactants are related by the stoichiometric equation for the reaction, where rate is equal to negative delta A by delta T is equal to negative delta B by delta T is equal to delta P by delta T. The negative signs for the changes in concentration of A and P indicates that A and B are being used up in the reaction while P is being produced. It has been established that the rate of reaction at a given time is proportional to the product of the concentrations of the reactants raised to the appropriate powers. The negative signs for the changes in concentration of A and B indicate that A and B are being used up in the reaction, B is being produced. It has been established that the rate of reaction at a given time is proportional to the product of the concentrations of the reactants raised to the appropriate powers. So rate is proportional to concentration of A raised to F and concentration of B raised to G. So rate K is equal to as an equation, rate is equal to K into concentration of A raised to F and concentration of B raised to G, where K is the proportionality constant called the rate constant. The components F and G must be determined experimentally. They are not necessarily equal to the coefficients of the balanced equation. The experiments in the rate equation are usually small whole numbers. The exponents in the rate equation are usually small whole numbers such as 1 or 2. There are also some cases in which the exponent 0 occurs. The values of the exponents are related to the number of molecules involved in the detailed steps that constitute the mechanism. The overall order of the reaction is the sum of all exponents. If, for example, the rate of reaction A to B is given by the rate equation, rate is equal to K into concentration of A raised to 1, where K is the rate constant and the exponent of the concentration of A is 1, then the reaction is first order with respect to A and first order overall. If the rate of reaction A plus B turns to C plus D is given by rate is equal to K of A raised to 1 and B raised to 1, where K is the rate constant and exponent of the concentration of A is 1, the exponent for the concentration of B is 1, and the reaction is said to be first order with respect to A, first order with respect to B, and second order overall. Many common reactions are first or second order. The possibility exists that exponents in a rate equation may be equal to 0 and the rate of reaction A to B is equal to, given by the equation, rate is equal to K of concentration of A raised to 0 is equal to K. Such a reaction is called a zero order and its rate, which is constant, depends not on concentrations of reactants but on other factors such as the presence of catalysts. Enzyme catalyzed reactions can exhibit zero order kinetics and the concentrations of reactants are so high. Enzyme catalyzed reactions can exhibit zero order kinetics when the concentrations of reactants are so high that the enzyme is completely saturated with reactant molecules. So, measures of enzyme activity. Enzyme activity may be expressed in a number of ways. There are two standard units to express enzyme activity. First is the enzyme unit, the amount of enzyme causing transformation of 1 mole of substrate per minute at 25 degree under normal conditions of measurement. The ketal or cat. The ketal is the accepted SI unit of enzyme activity. 1 ketal indicates the transformation of 1 mole of substrate per second. What is the accepted SI unit of enzyme activity? 1 ketal. Now let's learn about the Michaelis-Menten approach to enzyme kinetics. A particularly useful model for the kinetics of enzyme catalyzed reactions was devised in 1913 by Leonor Michaelis and Menten. A typical reaction might be the conversion of some substrates S to a product P. The stoichiometric equation for the reaction is S to P. The mechanism for an enzyme catalyzed reaction can be summarized in the form E plus S gives ES, then E plus P, K1, K2 and K minus 1. In this equation, K1 is the rate constant for the formation of the enzyme substrate complex. In this equation, K1 is the rate constant for the formation of the enzyme substrate complex ES from the enzyme E and the substrate S. K minus 1 is the rate constant for the reverse reaction dissociation of the ES complex to free enzyme and substrate, and K2 is the rate constant for the conversion of the ES complex to product P and subsequent release of product from the enzyme. When we measure the rate, so-called velocity of an enzyme reaction at varying substrate concentrations, we see that the rate depends on the substrate concentration. At relatively low concentration of substrate, initial velocity increases almost linearly with increase in the substrate. The release reaches a maximum velocity in increase in substrate concentration, and it doesn't increase any further by increasing the concentration of substrate. Concentration of substrate at which reaction velocity reaches half its maximum velocity is called Michaelis constant. So concentration of substrate at which reaction velocity reaches half its maximum value is called Michaelis constant or Km. Km is expressed as moles of substrate per liter. For example, Km is equal to K minus 1 plus K1 by Km is equal to K minus 1 plus K2 by K1. Lower the value of Km, the greater the affinity of the enzyme for the substrate. Lower the value of Km, the greater the affinity of the enzyme for the substrate. When Km is lower, the enzyme substrate affinity will be higher. In Michaelis-Menten equation, reaction velocity versus substrate concentration shows the hyperbolic plot. Km is the substrate concentration at which the enzyme has half maximal velocity. The saturation effect was first observed in 1930 when Michaelis and Menten studied the reaction in which the enzyme invertase hydrolyzes sucrose, a disaccharide, into monosaccharide, glucose and fructose. So sucrose is being reacted by invertase forming glucose and fructose. Michaelis and Menten noted two significant features about the kinetics of this reaction. When the substrate concentration was kept constant. So invertase acts upon sucrose, forming it glucose and fructose. Michaelis and Menten noted sucrose transforms to glucose and fructose with the help of invertase. Michaelis and Menten noted two significant features about the kinetics of this reaction. When the substrate concentration was kept constant and the enzyme concentration was increased, the rate of reaction increased. But when the enzyme concentration was kept constant and the substrate concentration increased, the reaction rate increased to maximum value and then leveled off. This is caused by the saturation of all the available enzyme binding sites for substrate molecules. So Km will be here, and then substrate concentration will be represented on the x-axis. Vmax volume is represented on the velocity is represented on the y-axis. Vmax by 2 then gives the Km. Then Vmax is also there. Michaelis and Menten put forward a mathematical equation to establish the mathematical relationship among the equations E, S and Vmax and Km. V is equal to Vmax of S, concentration of S by Km plus S, where Vmax and Km both are constants. Vmax, concentration of S by Km plus concentration of S, where Vmax and Km both are constant. Where concentration of S is very lower than Km, then V is proportional to concentration of S. That means when Km is higher than concentration of S, then V is proportional to concentration of S. When S is greater than Km, then V is equal to Vmax. When S is equal to Km, V is equal to Vmax by 2. If Km is very great than concentration of S, then V is proportional to concentration of S. If S is equal to Km, then V is equal to Vmax by 2. But when S is greater than Km, then V is equal to Vmax. We know from equation Km is equal to Kn minus 1 plus K2 by K1. For Michaelis-Menten equation, K2 is rate limiting. Thus, K minus 1 is greater than K2 and Km reduces to K minus 1 by K1, which is defined as the dissociation constant Ks for the enzyme substrate complex. The following assumptions are made in driving the Michaelis-Menten rate equation. The concentrations of substrate S is much greater than the concentrations of enzyme. The rate of formation of ES is equal to that of the breakdown of ES, the steady state assumption. Very little accumulation of P, so formation of enzyme substrate complex from E plus P is negligible. Steady state assumptions. The steady state hypothesis states that the concentration of enzyme substrate complex remains constant through much of the reaction. When enzyme and substrate are first mixed, the concentration will rise rapidly from 2 to a so-called already steady state level as illustrated in the figure below. Like concentrations are represented on the y-axis, time will be there, enzyme will be there, and E plus S can be seen. P will be there, S will be there, and E will be there. For a simple enzyme catalyzed reaction, E plus S gives ES, then it transforms to E plus P. The graph shows how the concentration of substrate, free enzyme, and enzyme substrate complex and product vary with time. What is happening in the case of product? It's increasing, but at the same time, concentration of substrate is decreasing. Enzyme concentration is always the same, and the enzyme substrate concentration increases. ES is almost constant. After a very brief initial period, enzyme substrate concentration reaches a steady state in which ES is consumed approximately as rapidly as it is formed. So dES by dt is 0. The amount of E and ES are greatly exaggerated for clarity. So dES by dt approximately 0. The amount of E and ES are greatly exaggerated for clarity. Note that E plus ES gives ET for total enzyme concentration, and that ES actually falls very slowly as substrate is consumed while E actually rises. Now let's learn about the Lineweaver-Burke plot or double reciprocal plot. It is very difficult to determine the Vmax directly from a plot of V against S, and therefore Km cannot be directly be determined in this way either. To overcome these difficulties of Michaelis-Menten equation can be rearranged in a number of ways to convenient graphical representation. If an enzyme obeys Michaelis-Menten kinetics, a plot of the reciprocal of the reaction velocity 1 by V as a function of the reciprocal of the substrate concentration 1 by S will fit a straight line. The slope of the line is Km by Vmax. The intercept on the vertical axis is 1 by Vmax. The intercept at the horizontal axis is minus 1 by Vmax. What is the slope of the line? Km by Vmax. Slope of the line is Km by Vmax. So 1 by V, 1 by S, concentration of S, then 1 by Vmax, slope Km by Vmax. X-intercept will be minus 1 by Km. Minus 1 by Km. A second widely used linear form of the Michaelis-Menten equation is the Eddy-Hofstee plot. So V by concentration of S gives minus V by Km plus Vmax by Km. A plot of V by concentration of S versus V has a V by concentration of S intercept of Vmax by Km, a V intercept of Vmax, and a slope of minus 1 by Km. So V by concentration of S will be on the y-axis, V on the x-axis. Then Vmax by Km, intercept and slope will be minus 1 by Km, and Vmax will be there. The significance of Kh and Vmax. The Michaelis constant Km, the Michaelis constant Km varies considerably from one enzyme to another and also with the different substrates for the same enzyme. By definition, this is equal to the substrate concentration at half the maximum rate. It is equal to the substrate concentration at half the maximum rate. In another way, Km represents the substrate concentration at which half the enzyme active sites are filled by substrate molecule. The value of Km is sometimes equated with the dissociation constant of enzyme substrate complex, ES. The larger the Km, the weaker the binding. This can be seen from equation 1.2. However, it is true only when K minus 1 is greater than K2, so that Km is equal to K minus 1 by K1. Km is equal to K minus 1 by K1. In general, Km must be expressed in terms of 3 rate constant. Nevertheless, Km in units of molarity is customarily reported together with other kinetic parameters or enzyme catalyzed reactions. To begin with, it is a quality that can be measured easily and directly. Furthermore, Km depends on temperature, the nature of the substrate, pH, ionic strength, and other reaction conditions. Therefore, its value serves to characterize a particular enzyme substrate system under specific conditions. Any variation in Km for the same enzyme and substrate is often an indication of the presence of an inhibitor or activator. The maximum rate Vmax has a well-defined meaning, both theoretically and empirically. It represents the maximum rate attainable, that is, the rate at which the total enzyme concentration is present at the enzyme substrate complex. Next is the turnover number. Turnover number of an enzyme is the number of substrate molecules converted to product by an enzyme molecule in a unit time when the enzyme is fully saturated with substrate. Its unit is second inverse. So K cat is equal to Vmax by Et. Et is equal to total concentration of enzyme. An enzyme with a high Km requires a higher substrate concentration to achieve a given reaction velocity that an enzyme with a low Km by the same turnover number. K cat by Km ratio is called specificity constants, is often thought of as a measure of enzyme efficiency. Note that either a large value of K cat, which means rapid turnover, or a small value of Km, which means high affinity for substrate, will make K cat or Km large, that is high specificity. K cat is equal to Vmax by Et. Significance of K cat and K cat by Km. Kcat is the turnover number for the enzyme at very low substrate concentration, where concentration of substrate is very low than Km. Most of the enzyme is free. Thus we can think of E is equal to E0, where E0 is the total concentration of enzyme. So that the Michaelis-Menten equation becomes V is equal to Kcat by Km E into S. Thus, the ratio Kcat by Km is equivalent to the rate constant for the reaction between free enzyme and free substrate. A comparison of Kcat by Km for the same enzyme with different substrates or for two enzymes with their different substrates is widely used as a measure of enzyme effectiveness. Next, turnover number and specificity constants for some enzymes which are given. Catalase, carbonic anhydrase, urease, etc. are given. Now, effect of temperature and pH. Since most of the enzymes are proteins, so they unfold and denature at high temperature. This denaturation causes a decrease or loss of catalytic activity, which in turn decreases the rate of reaction. Similarly, low temperature also inactivates the enzyme. An enzyme catalyzed reaction achieves its maximum reaction at an intermediate temperature called optimum temperature, about 40 degrees Celsius. However, enzymes like Taq DNA polymerase present in thermophilic bacteria Thermus aquaticus has optimal temperature of 72 degrees Celsius. Taq DNA polymerase. Effect of pH. Each enzyme has its optimal pH at which rapid activity occurs. For example, optimal pH for pepsin, protein digestive enzyme, is 2 and trypsin it is 8.5. Enzyme inhibition. Inhibition of enzyme activity can be irreversible or reversible. Irreversible inhibitors usually bound covalently to the enzyme. Many natural toxins like penicillin, physostigmine are irreversible enzyme inhibitors. In a reversible inhibition, the inhibitor can dissociate from the enzyme. Two most common types of reversible inhibition can competitive and uncompetitive inhibition. Examples of irreversible enzyme inhibitors: cyanides react with enzyme metal ions, respiratory chain enzymes, and primary targets. Disopropyl fluorophosphate (DFP) inhibits enzymes with active site serine, including acetylcholinesterase. Then sarin, nerve gas like DFP, and physostigmine and parathion, that is an insecticide, but especially inhibitory to insect acetylcholinesterase. Penicillin is also an inhibitor. Those are the irreversible type of enzyme inhibitors. First is the competitive inhibition. The structure of a competitive inhibitor closely resembles that of an enzyme's normal substrate. Because of its structure, a competitive inhibitor binds reversibly to the enzyme's active site. The inhibitor forms an enzyme inhibitor complex that is equivalent to the enzyme substrate complex. The effect of a competitive inhibitor on activity is reversed by increasing the concentration of substrate. At high concentration of substrate, all the active sites are filled with substrate and reaction velocity reaches the value observed without an inhibitor. So, E plus S gives ES and E plus P. But at the same time, there is happening a process where E plus I is happening. So, that rate is called K1. And EI or enzyme inhibitor complex is formed. So, K1 is equal to EI, concentration of E, into concentration of I by concentration of EI. In the presence of competitive inhibitor, the Michaelis-Menten equation becomes V0 is equal to Vmax into S by alpha Km plus S, where alpha is equal to 1 plus I by Ki. V0 is equal to Vmax, concentration of S, by alpha Km plus concentration of S, where alpha is equal to 1 plus I by Ki. The double reciprocal form of the equation will be 1 by V0 is equal to alpha Km by Vmax into 1 by S plus V 1 by Vmax. In competitive inhibition, Vmax stays constant and Km increases, but the inhibitor does not affect the turnover number of the enzyme. So when we draw the 1 by concentration of S and 1 by V0, with competitive inhibitor alpha is equal to 4, alpha is equal to 2, non-inhibitor alpha is equal to 1. So 1 by Vmax will be there, minus 1 by alpha Km will be the x-intercept. With the competitive inhibitor, without inhibitor. Without inhibitor, there will be a straight line as mentioned earlier. Michaelis-Menten plot of uninhibited enzyme activity versus competitive inhibition. Clinical treatment of methanol poisoning is a classical example of the exploitation of competitive inhibitory mechanism. In the case of methanol poisoning, methanol in the body is converted to harmful formaldehyde by alcohol dehydrogenase. A high dose of ethanol is used to await the effect of methanol because ethanol competitively binds with the active site of the alcohol dehydrogenase. So methanol poisoning is happening, so we will be giving ethanol. Okay. Next is the uncompetitive inhibition. First one was the competitive inhibition. There is a competition of inhibitor to bind with the active site. Next is the uncompetitive inhibition. In uncompetitive inhibition, rarely encountered, an inhibitor also binds at a site distinct from the substrate. However, an uncompetitive inhibitor will bind only to the enzyme substrate complex. On the other hand, non-competitive inhibitor binds to either the free enzyme or the enzyme substrate complex. In uncompetitive inhibition, apparent Vmax and Km both decreases. So enzyme plus substrate gives enzyme substrate that turn to enzyme plus product. Along with enzyme substrate complex, there will be an interaction with inhibitor. That rate is called K1. So ESI will be formed. So ESI is equal to concentration of ESI. In the presence of uncompetitive inhibitor, the Michaelis-Menten equation becomes V0 is equal to Vmax concentration of S by Km plus alpha S, where alpha is equal to 1 by I minus K1. The double reciprocal form of equation will be 1 by V0 is equal to Vmax by Km plus Vmax by Km, that is happening, and 1 by concentration of S plus alpha Vmax, alpha by Vmax. And what is happening in the graph? 1 by V0 will be there and 1 by concentration of S will be there, but no inhibitor with uncompetitive inhibitor, without inhibitor. Michaelis-Menten plot of uninhibited enzyme activity versus uncompetitive inhibition. Next is the non-competitive inhibition, mixed inhibition. Next is the non-competitive inhibition. The inhibitor binds to the enzyme at a site other than the active site. Inhibitor binding alters the enzyme's three-dimensional configuration and blocks the reaction. Inhibitor binds to either free enzyme or is complex. Non-competitive inhibition is not reversible by increasing the concentration of the substrate. The non-competitive inhibition Vmax decreases and Km stays constant. So in non-competitive inhibition with non-competitive inhibitor or non-competitive, no competitor, so 1 by V is there, 1 by concentration of S will be there, alpha Vmax will be there. So what is happening here? E plus S and ES and EP formation is happening. Along with that, enzyme combines with inhibitor and forms the enzyme inhibitor complex. But along with that, enzyme substrate complex is also forming inhibitor related complex. So K1 is equal to K of concentration of K into concentration of I by concentration of EI, and K1 dash is equal to another one, concentration of ES and concentration of I by concentration of ESI. In the presence of non-competitive inhibitor, the Michaelis-Menten equation becomes V is equal to Vmax concentration of S by alpha Km plus alpha dash S. The double reciprocal form of equation will be 1 by V0 is equal to alpha Km by Vmax into 1 by S plus alpha by Vmax. Effects of inhibitors on the parameters of the Michaelis-Menten equation. Type of inhibition, no inhibition, Vmax will be there and Km will be there. Competitive, Vmax will be there, alpha Km will be there. Uncompetitive, Vmax by alpha dash will be there, Km by alpha dash will be there. Mixed, Vmax by alpha dash will be there and alpha Km by alpha dash will be there. Next is the regulatory enzymes. Enzymes are regulated at two levels, one at gene level. Enzymes are regulated at two levels, one at the gene level, that is the enzyme synthesis, by change in the amount of enzyme synthesis, and other at the level of catalysis, by control of enzyme activity through conformational or structural alterations without change in total amount of enzyme synthesized. Regulatory enzymes exhibit decrease or increased catalytic activity in response to certain signals. Activities of regulatory enzymes can be modulated through reversible or non-covalent binding. Activities of regulatory enzymes can be modulated through reversible non-covalent binding by regulatory compounds or by reversible covalent modification. Next are the allosteric enzymes. Allosteric enzymes are a class of regulatory enzymes which increase or decrease catalytic activities in response to certain signals. Allosteric enzymes function through reversible non-covalent binding of regulatory compounds called modulators or effectors. The modulators for allosteric enzymes may be inhibitory or stimulatory. Often the modulator is the substrate itself. Regulatory enzymes for which substrate and modulator are identical are called homotrophic. When the modulator is the molecule other than the substrate, the enzyme is said to be heterotropic. Most allosteric enzymes are oligomeric, consisting of multiple subunits. Allosteric enzymes may have two functionally different binding sites. One of the site, the active site, binds the substrate and catalyzes the reaction. The other site is known as the allosteric or regulatory site, binds to the molecule, which is called effector or modulator. Such enzymes are known as allosteric enzymes. These enzymes are usually oligomeric in nature. What is oligomeric type? When the modulator is a substrate itself, regulatory enzymes for which substrate and modulator are identical are called homotropic. When the modulator is a molecule other than a substrate, the enzyme is said to be heterotropic. These enzymes are usually oligomeric in nature. Effector molecules are of two types, namely positive effectors called activators that enhance enzyme activity, and negative effectors or inhibitors which inhibit enzyme activity. Binding the effectors causes conformational changes in the allosteric enzymes which influence their catalytic activity. Enzymes with several modulators generally have different specific binding sites for each. In homotropic enzymes, the active site and regulatory site are the same. Allosteric enzymes don't obey Michaelis-Menten kinetics. Allosteric enzymes show relationship between V0 and S that differ from Michaelis-Menten kinetics. They do exhibit saturation with the substrate when S is sufficiently high, produce a sigmoid saturation rather than the hyperbolic curve typical of non-regulatory enzymes. Sigmoid kinetic behavior generally reflects cooperative interactions between protein subunits. The principles are particularly well illustrated by the known enzyme O2 binding to hemoglobin. Sigmoid kinetic behavior is explained by the concerned and sequential model for subunit interaction. Vmax will be there, half Vmax will be there, then the VO will be there, and S and all other types are given. So the sigmoid curve of a homotropic allosteric enzyme is there. The effect of a positive modulator and a negative modulator in heterotropic allosteric enzyme. The central curve shows the substrate activity relationship without a modulator. Next, some enzymes and proteins whose activity can be controlled by reversible covalent modification. Glycogen phosphorylase, glycogen synthetase, acetyl-CoA carboxylase, glutamine synthetase, nitrogenase, phosphorylation, and adenylation and ADP ribosylation are taking place. Now let's learn about the isozymes. Multiple forms of an enzyme that catalyze the same reaction but differ from each other in their amino acid sequence, composition, substrate affinity, Vmax, and regulatory properties are described as isozymes. A common example is the lactate dehydrogenase (LDH), which catalyzes the reversible conversion of pyruvate to lactate. There are 5 LDH isozymes. Each is a tetramer of two different types of subunits called H and M. Two different subunits combine randomly with each other, forming 5 isozymes. Next is zymogen. An inactive precursor of an enzyme is called zymogen. Zymogen is cleaved to the active enzyme. Many proteolytic enzymes like chymotrypsin, trypsin are initially synthesized as inactive precursor chymotrypsinogen and trypsinogen. Specific cleavage causes conformational changes in the inactive precursor that exposes the enzyme active site. In the duodenum, the pancreatic zymogens, trypsinogen, chymotrypsinogen, proelastase, and procarboxypeptidase are converted into active enzymes by enteropeptidases and trypsin. So, pepsinogen, trypsin, and chymotrypsinogen, enteropeptidase, elastase, carboxypeptidase, activation of pancreatic zymogens. Next is the chymotrypsinogen. A single polypeptide chain of 245 amino acid residues is converted to alpha chymotrypsin, which has 5 peptide chains linked by 2 of 5 disulfide bonds present in the primary structure of chymotrypsinogen. Chymotrypsinogen is inactive. Chymotrypsin is active. by chymotrypsin and alpha chymotrypsin. It is an active form. Formation of chymotrypsin active from the chymotrypsinogen inactive. The numbering of amino acid residues represents their positions in the primary sequence of the zymogen chymotrypsinogen. The three polypeptide chains of chymotrypsin are linked by disulfide bonds. Next, ribosome. A ribosome from ribonucleic acid enzyme, also called RNA enzyme, is an RNA molecule that catalyzes a chemical reaction. Many natural ribosomes catalyze either their own cleavage or the cleavage of their RNAs, but they have also been found to catalyze the aminotransferase activity of the ribosome. Before the discovery of ribozymes, proteins were the only known biological catalysts. In 1967, Carl Weiss, Francis Crick, and Leslie Orgel were the first to suggest that the RNA could act as a catalyst based upon findings that it can form complex secondary structure. The first ribosome was discovered in 1980s by Thomas R. Sesh, who was studying RNA splicing in the ciliated protozoan Tetrahymena termophila. This ribosome was found in the intron of the RNA transcript and removed itself from the transcript. Ribozymes often have divalent metal ions such as Mg2+ as cofactors. In 1989, Thomas R. Sesh and Sidney Altman won the Nobel Prize in Chemistry for their discovery of catalytic properties of RNA. Although ribozymes are quite rare in the cell, their roles are sometimes essential for life. For example, the functional part of the ribosome, the molecular machine that translates RNA into proteins, is fundamentally a ribosome. Some known ribozymes include RNase P group 1, group 2 introns, hairpin ribozymes, hammerhead ribozymes, hepatic delta virus ribosome, and tetrahymena ribosome. If ribozymes were the first molecular machines used by early life, then today's remaining ribozymes, such as the ribozyme machinery, could be considered living fossils of a life based primarily on nucleic acids. Example of enzymatic reactions. Lysozyme is an enzyme present in the number of secretions such as tears, sweat, and also found in the egg white. Alexander Fleming, who discovered penicillin, described a lysozyme in 1922. Its structure was described by David Phillips in 1965. Lysozyme, present in the hen egg white, is a monomer with 129 amino acid residues. It cleaves the beta-1,4 glycosidic bond that connects N-acetyl muramic acid with the fourth carbon atom of N-acetyl glucosamine of polypeptidoglycan cell wall. It does this by binding to the peptidoglycan molecules in the binding site. The reaction catalyzed by lysozyme is a hydrolysis. A molecule of water is added to the single bond between two adjacent sugar groups in the polysaccharide chain. Now let's learn about vitamins. Vitamins are organic compounds required by the body in trace amounts to perform specific cellular functions. They can be classified according to their solubility and their functions in metabolism. Vitamins cannot be synthesized by humans and therefore must be supplied by the diet. What are soluble vitamins? 9 vitamins like thiamine, riboflavin, niacin, biotin, pantothenic acid, folic acid, cobalamin, pyridoxine, and ascorbic acid are classified as water soluble, whereas 4 vitamins, vitamin A, D, E, K, are termed fat soluble. First is the thiamine, like vitamin B1. Thiamine pyrophosphate, TPP, is a biologically active form of vitamin formed by the transfer of a pyrophosphate group from ATP to thiamine. TPP serves as a coenzyme in the oxidative decarboxylation of alpha keto acid and the formation of degradation of alpha ketones by transketolase. Beriberi is a severe thiamine deficiency syndrome found in areas where polished rice is the major component of the diet. So that is something which is related with thiamine. Thiamine, oxidative phosphorylation of keto acid and the formation of degradation of alpha ketones by transketolases, beriberi disease. Riboflavin is the vitamin B2. The two biologically active forms are flavin mononucleotide and flavin adenine dinucleotide. FMN is synthesized after the addition of phosphate in riboflavin, and FAD formed by the transfer of an AMP moiety from ATP to FMN. FMN and FAD are each capable of reversibly accepting two hydrogen atoms, forming FMNH2 or FADH2. Flavin molecule, riboflavin, that is vitamin B2. Next is niacin. Niacin or nicotinic acid is a substituted pyridine derivative. The biologically active coenzyme forms are nicotinamide adenine dinucleotide NAD+, and its phosphorylated derivative nicotinamide adenine dinucleotide phosphate NAD+, P+. That is niacin. Nicotinamide, a derivative of nicotinic acid that contains an amide instead of a carboxyl group. NAD+ and NADP+ serve as cofactors, coenzymes in oxidation reduction reactions in which the coenzyme undergoes reduction of the pyridine ring by accepting a hydride ion. The reduced forms of NAD+ and NADP+ and NADH and NADPH respectively. Deficiency of niacin causes pellagra, a disease involved the skin and central nervous system. The symptoms of pellagra progress through the 3Ds like dermatitis, diarrhea, dementia, and if untreated, death. That is niacin pellagra disease. The next is the biotin. Biotin is a coenzyme in carboxylation reactions in which it serves as a carrier of activated carbon oxide. Biotin is covalently bound to the enzyme by an amide linkage between a carboxyl group of its valerate side chain and the alpha amino group of an enzyme residue to form a biocytin. Alternatively, biotinyl lysine residues. Biotin deficiency does not occur naturally because the vitamin is widely distributed in food. Raw egg white contains a glycoprotein avidin which tightly binds biotin and prevents its absorption from the intestine. Raw egg white contains a glycoprotein avidin which tightly binds biotin and prevents its absorption from the intestine. So that's why we shouldn't consume raw egg white in any form, because that blocks the absorption of biotin. Then the pantothenic acid. Pantothenic acid is a component of coenzyme A which is responsible for the transfer of acyl groups. Coenzyme A contains a thiol group and carries acyl components as activated thiol esters. Next is the folic acid. Folic acid or folate plays a key role in transfer of one carbon unit and is essential for the biosynthesis of certain amino acid, the purines and thymidylate. Folic acid is composed of a pterin ring attached to p-aminobenzoic acid and conjugated with one or more glutamic acid residues. The biologically active form of folic acid is tetrahydrofolic acid. NADPH dependent enzyme dihydrofolate reductase and catalyze reduction of 5, 6, 7 and 8 positions of ring. The primary result of folic acid deficiency is megaloblastic anemia caused by diminished synthesis of purines and pyrimidine, leading to the inability of cells to make DNA and to divide. Megaloblastic anemia. Folic acid deficiency causes megaloblastic anemia. Then next is the cobalamin or vitamin B12. Vitamin B12 is required in humans for two essential enzymatic reactions, synthesis of methionine and the isomerization of methyl malonyl CoA, which arises from fatty acids and odd numbers of carbon atoms. Corindin is there. And next is the pyridoxine. Pyridoxine or vitamin B6. Vitamin B6 is a collective term for pyridoxine, pyridoxal and pyridoxamine, all derivative of pyridine. Next is the ascorbic acid. Vitamin C is a well documented, very important reaction in hydroxylation reaction, antioxidant by virtue of its water solubility. Deficiency of vitamin C causes scurvy, a disease characterized by sore, spongy gums, loose teeth, fragile blood vessels, swollen joints, and anemia. Many of the deficiency symptoms can be explained by the deficiency of the hydroxylation of collagen, resulting in the defective connective tissue. Fat soluble vitamins will be explained later.